Welcome to another video class from Karims Biology. In today's class, I am going to teach you about cell organelles, ribosomes, lysosomes and vacuoles. We are going to discuss about their structure and also about their functions. Okay, so let us start the session.
Subscribe now and press the bell icon. Never miss an update.
First of all, let us learn about ribosomes. Ribosomes are the single-walled spherical bodies composed mainly of RNA. RNA stands for ribonucleic acid, and in addition to RNA they are also composed of some associated proteins. So this is the ribosome, and when I taught you about endoplasmic reticulum, I told you that the surface of SER is studded with some granule-like parts called ribosomes. So this is that granule-like particle which is attached to the surface of RER, which is rough ER. And what is it made up of? This ribosome is made up of RNA. RNA is a type of nucleic acid like that of DNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. So it is made up of mainly RNA called ribosomal RNA and some associated proteins. Together they form a spherical body called ribosome. And in this picture of ribosome, you can see there is a large part here and there is another small part over here which is illustrated with some other color. So this makes it clear that the ribosome consists of two parts which are usually called subunits. Yes, so this is the large subunit which is bigger in size, and the other one is the smaller subunit. So the ribosome has two ribosomal subunits. The first one is the larger subunit and the second one is the smaller subunit. Here the size of this ribosome is appearing to be bigger, but in fact it is very small, which is attached to the surface of the endoplasmic reticulum, and some ribosomes are freely suspended in the cytoplasm. And there are two major types of ribosomes which are distributed among prokaryotic cells and eukaryotic cells. These two major types are called 70S ribosomes and 80S type of ribosomes. 70S means Svedberg unit. Okay, so details of these types of ribosomes are not required for your class. You are going to learn about these types of different ribosomes in your higher classes. Okay, now let us discuss about location. I told you already that ribosomes, they are present in two different places within the cell. They are the small granule-like structures either scattered in the cytoplasm. Okay, they may be freely suspended within the cytoplasm. They are not attached to anything. Freely they are floating in the cytoplasm. They are called free ribosomes. Why are they called free ribosomes? Because they are not attached to any part of the cytoplasm. That's why they are called free ribosome. Or the ribosomes may also attach to endoplasmic reticulum. As I have told you in the previous session that there will be endoplasmic reticulum. Okay, and endoplasmic reticulum is formed by the protrusion of outermost layer of the nucleus. So this is ER, and I have told you that there are two types of ERs. One is rough ER, other one is the smooth ER. And I have mentioned that to the surface of rough ER, these granule-like structures are attached. So this type of ribosomes are called bound ribosomes. Right? So that's about the types of ribosomes. Now come to the function. What is the function of ribosomes? The major function of ribosomes is protein synthesis. I told you that endoplasmic reticulum, particularly rough ER, is associated with ribosomes because the ribosomes are attached to the surface of rough ER. And do you remember the function of rough ER? I told you that rough ER is responsible for protein synthesis within the cell. And rough ER is able to do that because these ribosomes are attached to the surface of ER. In other words, for the protein synthesis in the cell, ribosomes plays a crucial role. Okay, that's why most often the ribosomes are referred as protein factories of the cell. Okay, so that is the only major function of ribosomes. Right? So that's all about ribosomes. Now let us know about lysosomes. What are these lysosomes? Lysosomes are membrane-bound sac-like structures that are budded off from the Golgi complex. So this too I have taught you when I discussed about the functions of Golgi complex. Do you remember Golgi complex? Yes, this is the ER, rough ER. The rough ER prepares the proteins, and I have told you that from rough ER, small buds will be pinched off, which are called transport vesicles. They consist of some unprocessed proteins. These transport vesicles will go and fuse with the cisternae of the Golgi apparatus. This is how the proteins manufactured in the ER are transported to Golgi apparatus. Within Golgi apparatus, the proteins are modified, vigorously processed, and then they will be made ready for the use within the cell. The processed proteins or modified proteins now will be sent to different parts of the cell wherever they are required with the help of secretory vesicles. Okay, and I told you that some of the secretory vesicles, they turn into lysosomes because they consist of some proteins which act as enzymes. Don't forget this point. All enzymes are proteins. All enzymes are nothing but proteins. So here lysosomes are filled with proteins, and what type of proteins are present in lysosomes is very important. They are called hydrolytic enzymes. So what is present within the lysosome? The lysosome consists of hydrolytic enzymes. So this is how ribosomes are formed, and ribosomes are small bag-like structures, small sac-like structures. Don't forget this point. They are membrane bound. They are surrounded with membrane, and they are sac-like structures, small bag-like structures which are filled with hydrolytic enzymes. Now come to the point. What are these hydrolytic enzymes? Hydrolytic enzymes are destructive enzymes which are able to destroy any biological molecule, or which are able to digest any of the macrobiological molecule. That is the importance of these hydrolytic enzymes, and as they are able to destroy anything, as they are able to destroy even the cell organelles present in the cytoplasm of the cell, they are able to destroy any foreign particle that enters into the cell. That's why the hydrolytic enzymes are called destructive enzymes. Now let us see the location of these lysosomes. Lysosomes are mostly found in the cytoplasm of animal cells. So this is an important point to be remembered. Okay, so where do you find lysosomes? Mostly they are found in the cytoplasm of animal cells, but recent studies indicated that even plant cells consist of a few of the lysosomes. But in animal cells only, lysosomes play a crucial role in digestion process, particularly in intracellular digestion, and also for the protection of the cell. I will tell you how that happens within a couple of minutes. Okay, so that's about the location, and why in plant cells lysosomes are less in number means the function of lysosomes in plant cells, as per the recent study, the vacuole present in the plant cell, it does the function of lysosomes. That's why they don't play much role in the plant cell, whereas they play a crucial role in animal cells cytoplasm. And what is the role played by these lysosomes, we will be discussing. Okay, so now let us discuss the functions of the lysosomes. Okay, the first function of lysosome is intracellular digestion. What is it? Intracellular. Intra means within the cell. Within the cell, if the digestion happens, then that is called intracellular digestion. So I will explain the first point. Okay, now look at this picture. In this picture, this is the plasma membrane, this is the cell membrane of the cell, this is the cytoplasm, this is rough ER where the proteins are manufactured, this is Golgi apparatus where the proteins are modified and lysosomes are pinched off. The first point is the lysosomes involve or helps in intracellular digestion. So what is this intracellular digestion? You know, the cell takes its food from outside. Imagine this is a phagocyte, or imagine if it is a unicellular animal cell, and the unicellular animal cell has taken the food material. The food will be taken by a process called endocytosis. I hope that you remember, I taught you about endocytosis when we discussed about the functions of plasma membrane. Okay, so the food particle, when it comes into contact with the cell membrane grows inwards and the food particle will be taken into the cytoplasm. The cell membrane now pinch off and form a vacuole filled with food material which is taken from outside. So in this way, the food material here is the product which is entering into the cell from extracellular place. Extracellular means from outside. So this is the extracellular item which is entering into the cell. So once it enters into the cell, as it is a food particle, the food particle should be digested, right? The food particle should be digested, should be broken into small pieces, and that broken small particles will be used by the cells within the cytoplasm. Now how this food particle will be now broken down? It will be done with the help of lysosomes. Now you see what is happening here. The food has been taken into cytoplasm. The food formed the food vacuole. Now from the Golgi apparatus, a lysosome along with the hydrolytic enzymes or digestive enzymes is pinched off, and this lysosome is moving towards the food vacuole. Ultimately the lysosome is fusing with the food vacuole, so that the enzymes present in the lysosome, the digestive enzymes present in the lysosome, now they enter into food vacuole. When the digestive enzymes enter into food vacuole, the food will be broken into small pieces, and these simple molecules of food will now enter into cytoplasm, diffuse into cytoplasm, where they will be used for different processes. So this is how digestion of the food material occurs. But let me ask you one question. Where does the digestion occur here? The digestion here, it happened within the cytoplasm, that is within the cell. That is why it is called intracellular digestion. As the digestion of the food particle is occurring within the cell, it is called intracellular digestion. And what is the second important function of the lysosome? The second function is the lysosomes are able to destroy foreign substances such as pathogens. You know, from outside, from the extracellular space, not only food particles, sometimes even some of the pathogens such as bacteria can also enter into the cell. So there is a chance that from outside, even bacteria and some other pathogens, even some of the viruses also may enter into cytoplasm. In the same way, how the food is taken. When the bacteria enters into the cell, when it forms this small sac-like structure, it is called phagosome. Now what happens? The same lysosomes will come and fuse with this phagosome, releasing all these destructive enzymes into the phagosome. As a result, these enzymes now they digest the bacteria. They kill the bacteria and they digest the bacteria, and the digested contents of the bacteria will be sent out of the cell. Okay, so this is how the lysosomes are also helping for destruction of the foreign substance which enters into the cell. That may be a bacteria, that may be a spore, that may be a virus, whatever it may be, whichever comes from outside is the foreign material. Especially pathogens will be destroyed by these lysosomes. And the last one is when cell is old or injured. This is very important point. When cell is old or injured, these lysosomes rapidly destroy organelles and kills the cell. Hence they are called suicidal bags of the cell. So this is one of the very important role played by lysosome. What is it? The lysosomes, they are also able to destroy the worn out cells, or they are also able to destroy the cell organelles which are no longer useful within the cell. For example, you see here, imagine this is a lysosome, and can you see there is some yellow colored structure within this lysosomes here? Actually this is mitochondria. This is mitochondria. So I think you may be in a confusion. Why mitochondria is within the lysosome? Yes, the mitochondria here, it is within the lysosome because this mitochondria has been damaged. It is no longer functioning within the cell. It is worn out, it has become old. Okay, so that is another job of the lysosome. Imagine that there is a cell organelle here, or there is a mitochondria here which is worn out or which has become old. Now what happens when the mitochondria becomes old? Worn out mitochondria will be transported to lysosome. It enters into lysosome. After entry of this mitochondria into lysosome, the powerful destructive enzymes destroy this mitochondria. You can see here. Okay, the mitochondria will be destroyed. Not only mitochondria, whatever the cell organelle which is worn out or which is no longer working, which has become old, will be destroyed within the lysosome. That is another major function of lysosome. Okay, and sometimes when the entire cell becomes old, when all the cell organelles become old, or when the cell is infected with any pathogen which cannot be removed, then the cell commits suicide. When the cell is no longer useful, when all the cell organelles in the cell are infected with any microorganism, or when the cell is when the cell entirely has become old, then during some time the lysosomes present in the cell, they burst open. The lysosomes present in the cell, they burst open so that all the hydrolytic enzymes will come into cytoplasm. All the hydrolytic enzymes come into cytoplasm. When the hydrolytic enzymes come into cytoplasm, all the cell organelles present in the cytoplasm will be destroyed by these digestive enzymes or destructive enzymes. Okay, in other words, the cell is destroying itself. So nothing from outside is destroying the cell. The cell is destroying itself from within by using these lysosomes. Okay, so in other words, the cell in fact is committing suicide. And how this suicide is happening? It is happening by these lysosomes. The lysosomes are helping for that process. That is why most often the lysosomes are named as suicidal bags of the cell. Okay, so this is called cell death, when the cell destroys itself by bursting the lysosome and releasing the contents of lysosome into cytoplasm. Then that is called cell death, and for this cell death, lysosomes play an important role. Okay, that's why they are called suicidal bags of the cell. Usually, the lysosomes contains digestive enzymes. The digestive enzymes are packed within the lysosome, and there is a membrane separating the digestive enzymes to that of the cytoplasm. So as long as the digestive enzymes stay within the lysosome, the entire cell is safer. And when the lysosomes burst open and release all the hydrolytic enzymes into the cytoplasm, okay, then all the other cell organelles will be destroyed, which ultimately results in the death of the cell. This is called cell death, and this is a kind of suicide which is triggered by bursting of lysosomes. That's why they are called suicidal bags of the cell. Okay, so that's about lysosomes. Now let us discuss about vacuoles. First, let us know what are vacuoles. Vacuoles are the large empty spaces present in the cells. So this is a plant cell over here, and I hope that you remember, when you draw the structure of plant cell, at the middle of cytoplasm, we usually draw a large balloon-like structure, a large bag-like structure which almost occupies entire cytoplasm. This is a vacuole. Okay, so these are large empty spaces present in the cells called vacuoles, but they are not actually empty because they are fluid-filled sac-like structures, and they are filled with water and other substances. They will be filled with water, and they may also be filled with amino acids. Some of the sugars may be present, and of course the waste material may also be present within the vacuole. Okay, and proteins may be present in the vacuole. Okay, so that is the reason why we cannot say they are empty. They are fluid filled sac like structures with water and other important substances within the solution. Okay, and now let us see where are they present. They are present both in plant cells and animal cells, but there is a difference that you have to remember here. What is that? Plant cells have few vacuoles. Some of the plant cells, they have a single vacuole, but the size of the vacuole in plant cell is bigger. It is bigger in size. You can see here, this is the plant cell. It is bigger in size in plant cells, whereas in animal cells, this is the animal cell. In animal cell, you look at the size of the vacuole is very small comparative to the size of the vacuoles in plant cells. So this is another important difference between plant cell and animal cell. The plant cell consists of a large vacuole, whereas the animal cell consists of a small vacuole compared to the plant cells. Okay, now the outer covering of vacuole is called tonoplast. Okay, so this is the vacuole, and can you see this layer here? Yes, there is a membrane which is covering the vacuole. This outer membrane which is covering the vacuole, this is called tonoplast. This outer covering is called tonoplast, and the presence of tonoplast is actually separating the contents of the vacuole from the contents of the cytoplasm. What is present outside the vacuole? Outside of vacuole there is cytoplasm. Within the vacuole there is cell sap. Okay, so the contents of the vacuole together called cell sap. So that's about the size of vacuole as well as the outermost layer which is called tonoplast. Now let us come to the functions of vacuole. So the major function of the vacuole is storage of water and other substances such as wood materials, pigments, and most importantly, they also store some of the waste products within the vacuole. That is the reason why when the age of the plant cell becomes old, the size of the vacuole becomes bigger and bigger because it will be storing all the substances, especially waste materials will be stored in the vacuole. And another important function of vacuole is it gives turgidity to the cell to the plant cell by pressing against the cell wall. Children, I have told you previously that the cell wall is going to exert some pressure towards the cytoplasm, which is called inward wall pressure. And if the wall pressure is not compensated with opposite equal pressure, then the cell may collapse. So that is why the vacuole here plays a crucial role in maintaining the turgidity of the cell. So what the vacuole does here is, the vacuole, as it is filled with water, it is going to exert some outward pressure which is exactly opposite to the inward pressure exerted by the cell wall. So the cell wall is exerting this pressure towards inner side, whereas the contents of the vacuole is going to exert outward pressure, so that both inward wall pressure and outward pressure from the vacuole are going to be compensated. As a result, the cell become tough and turgid. Okay, so this is another important function served by vacuole within the plant cell. So these are the functions of vacuole. I hope that you understood. That's all for today, children. I will meet you in my next session. Thank you for watching.